Today, greeting from the Era Medical College, let us try to talk about a fourth pillar in the treatment of cancer. Up to now, the three pillars in the treatment of cancer have been RT, radiotherapy, chemotherapy, and surgery. But the normal human body, with about 14 trillion multiplications, cancer cells are being produced in each one of us every day, and they are recognized by the immune system and destroyed. However, this normal system, immune system that prevents, surveillance system that prevents cancer, detects cancer and destroys cancer cells, is rendered ineffective in cancer by the following mechanisms. Actually, if this is the cancer cell, this cancer cell is different genetically from a normal cell. Supposing this is the normal cell, in about 1300 genes. And these genes express as proteins which are exclusive to cancer cells. Most of the proteins lie in the nucleoplasm, some in the nuclear membrane, some in the cytoplasm. And of the 7 to 10 that present at the surface of these cells, only about 2 are correctly oriented so that they can be recognized by the immune system and destroyed. So, the first protection is that there are only about 2 or 3 antigens which are correctly oriented and can lead to recognition by the cell. The second thing is that these antigens that express on the surface, they stimulate B cells, and these B cells produce haptens, capping antibodies that keep capping these antigens, and thus the tumor cell is hidden from the immune system by these ineffective antibodies, and this is known as eclipse phenomenon. The tumor hides from the immune system. The third thing that happens is that these immune complexes, like this, they circulate in blood, and the T cells are they tend to go to sleep because of these immune complexes. And also, these same immune complexes can decrease their production also. So because of this, what has come recently is that these T cells, as they try to recognize the tumor cells, these T cells are sort of also fooled, and these T cells have 2 types of antibodies on them, like this. One tries to say self or non-self, and the other sort of kills. So these T cells, because of this self-non-self recognition, recognize the cancer cell as self and do not kill. So they do not kill. Now what has recently happened is that from the patient, you get T cells, and this receptor, one is the recognition system and the other is the killing system. So by a single chain variable fragment, you sort of stop the tolerance system, and if this cell will therefore only kill. So this this receptor with a single chain variable fragment, now it's a single chain variable fragment, is called as the CAR, chimeric antigen receptor. And these cells are called as CAR T cells. So from the patient you take the T cells and then you genetically engineer them by introducing some genes which are on the plasmid. Some genes which are on the plasmid, you introduce them using a retro or a lentivirus into the nucleus of the T cell, and therefore these T cells start expressing this receptor, and this receptor, this cell will not recognize tolerance. It will go and destroy the tumor cell which bears this antigen. So if this is, let us say, CD19, and CD19 is 70 on the tumor cell and only 20 on the normal cell, therefore 17 tumor cells will be destroyed and only 20 normal cells will be destroyed. So this makes it highly specific. Now these CAR T cells, when they have been used, there is another thing that goes with it. That once this gets attached, this CD19 gets attached to this receptor, this has got co-stimulation. The co-stimulation is by a brake, which is CTL4, and it has got a accelerator also. So if you have to release the brake and stamp on the accelerator for this to be more effective. So even after the antigen is bound to this CAR receptor, still you need an accelerator and a brake to cause an optimal response by these T cells. As far as this goes, then when these T cells are injected into the patient's body, they proliferate, they keep on destroying tumor cells, and when there are no tumor cells, they die down. They are finished. But if there is a big war on, there will be a lot of cytokine release. So there can be a cytokine release syndrome in tumors which have a large number of mass. And this can be prevented by IL-6 blockers, the drugs that block the interleukin 6. The second important thing is that if in the proliferation you use IL-2 and if you use lymphodepletion in hematological malignancies so that the tumor cell numbers are less, then the survival is more in case as shown by the meta-analysis. So these are the nuances of CAR T cell therapy, and basically you have greater than 90% tumor-free survival in hematological malignancies, and if this war is on in neural tissue, then there can be neurotoxicity. And if that ratio 20 CD19 on normal cell and 90 CD19 on tumor cell, so if that ratio is more 50-50, then there can be B cell aplasia also. So 90% tumor-free survival in hematological malignancies which are relapsed or recurred, relapsed or refractory. Another interesting fact about the CAR T cells now is that in solid tumors, T cells which have been cultured from the tumor and converted to CAR T cells are able to cross over the microenvironment of solid tumors and still kill and effectively kill tumor cells. So there has been a lot of advance in solid tumors as well. Now this is all.